1958, scientists Matt Meselson and Franklin Stahl began to ask the question, how does DNA replicate? At this time, there were three proposed models for DNA structure. The first was conservative, in which DNA replication results in one molecule that consists of both original DNA strands and another that consists of two new strands with the same sequences as the original molecule. Semi-conservative believes that replication results in two DNA molecules with one original strand and one new strand. The final model, dispersive, proposed that replication results in two DNA molecules that are mixtures of the parental and the daughter DNA. Meselson and Stahl decided to investigate and learn which model was accurate. In their experiments, they worked with E. coli. They started by growing E. coli in a container containing nitrogen-15, a heavy isotope of nitrogen. The E. coli used up the nitrogen and used it to create new biological molecules with new DNA. The bacteria was then moved to a container containing nitrogen-14, a light isotope of nitrogen, and given time to grow and replicate. Meselson and Stahl collected samples from each generation and measured the density of the DNA using a density gradient centrifuge. This separates molecules into bands based on density. This would allow the DNA with the nitrogen-15 isotope to separate from the DNA with the nitrogen-14 isotope. The results showed some very interesting data. DNA from cells at the start of the experiment produced only one band, as it only contained the heavy nitrogen. The first generation after being put into the nitrogen-14 isotope produced one band that was slightly higher because it had some of the light nitrogen mixed into the DNA. This showed that these molecules had a hybrid of the light and heavy nitrogen in the DNA. This ruled out the conservative model, but there were still two choices. Semi-conservative replication would have a double-stranded DNA with one strand of nitrogen-15 DNA and one strand of nitrogen-14 DNA, and dispersive would result in double-stranded DNA with both strands being a mixture of nitrogen-15 and nitrogen-14 DNA, both having an intermediate density. After the bacteria had replicated multiple times, they found equal amounts of DNA with the two densities, one having the intermediate density and the other having a lighter density from growing in the nitrogen-14 solution. This ruled out dispersive because there were two different densities and not only one that was a mix of the light and heavy nitrogen. The results of this experiment are extremely significant even still today, and it is now known that semi-conservative DNA replication is a universal mechanism that all organisms use to replicate DNA, and we can thank Meselson and Stahl for their discovery.